Hello everyone, and welcome to the fifth plenary lecture of the 2020 Goldschmidt Conference. I'm Jochen Brocks. I'm from the Research School of Earth Sciences of the Australian National University. And my co-author is Benny Nettersheim, my former PhD student, who is now at the MPI in Bremen. You are going to learn in this lecture something about your very earliest ancestors. You know, when I go into the 19th century, I have a great uncle who was the cultural minister under Otto von Bismarck in Prussia. And I like him very much. He's my most favorite ancestor because he invented the 5-minute school break. But I also have a great grandmother to the 7th degree. She was the Princess of Stolberg. But she blew my inheritance, the famous and enormous, beautiful Stolberg castle, because she ran away with the castle's chef. Now you will have your own illustrious, notorious ancestors, and generally we even seem to be obsessed with our ancestry. And some of us are even obsessed with our ancestry, going back in time hundreds of millions to billions of years. And there is someone who wrote a fabulous book just about that. In The Ancestor's Tale, Richard Dawkins describes a pilgrimage back to the dawn of life, describing our deeper and deeper ancestors. According to The Ancestor's Tale, we have common ancestors with the chimpanzee and the bonobo 5 million years ago, and with a gorilla 7 million years ago, followed by the orangutan, Old and New World monkeys, the prosimians, rodents, marsupials, and finally the monotramps. Now, amazingly, you share your grandparents to the 120 millionth degree with a platypus. But we don't have to stop here. We can go deeper back in time. Beyond the monotremes, there will be the sauropsids, which is the birds and the lizards, and then the amphibians, followed by lungfish, sharks, protostomes, and finally cnidarians, and then the sponges. That creature down here is called Laka, the last animal common ancestor. It lived probably roughly around 800 plus minus a few hundred million years ago. But we can go yet deeper back in time. Beyond Laka, we will finally eventually join the fungi, yet much deeper the amoebae, until we finally arrive at the last common ancestor with all other eukaryotic creatures, including the plants and algae. This creature here is now called Lika, the last eukaryotic common ancestor. It lived roughly, according to molecular clocks, around 1,200 to more than 2,000 million years ago. Lika and all its descendants are called the crown group eukaryotes because they form the crown of the eukaryotic tree. You know, eukaryotes are all those creatures, as opposed to bacteria and archaea, that have relatively large cells, that have a nucleus, that have organelles and a cytoskeleton. We are eukaryotes. Yet beyond Lika, yet deeper down in time, there was a lost world of complex life, of old world eukaryotes that we call stem group eukaryotes, all of them now extinct. Unfortunately, Lika is a genomic black hole. It is almost impossible to extrapolate information back in time from living creatures beyond the node of Lika. Therefore, to learn something about the eukaryotic stem, we have to go back in time into the geological record. This here is a time scale from 1.8 billion years ago to the Paleo, Meso, and Neoproterozoic, all the way to the present. And the creature, the fossil that you see down here, is the oldest eukaryotic fossil in the geological record, Valeria lophostriata. It has cell wall ornamentation, proving that this thing had a cytoskeleton and therefore it was eukaryote. But we do not know what sort of eukaryote. Now, these ancient eukaryotic fossils are not very common, but paleontologists keep finding them. But what these creatures were exactly, we do not know. The first eukaryotes that we can recognize as something that still exists today emerged around 1 billion years ago. And this is Nick Butterfield's fabulous and famous bengiomorph red alga. This is Qiang Tang's and Chuhai Zhao's recently discovered green algae. But both of these are singular occurrences, extremely rare. But crown group eukaryotes, our crown, really starts to explode around 800 million years ago, with the emergence of Quentin Laurence and Emmanuel Javot's fabulous fungi, Susanna Porter's rhizarians, including the group of foraminifera and amebozoans, Phoebe Cohen's beautiful phosphatic scales, which may have been part of a haptophyte alga, and even ciliates, discovered by Tanja Bozak. Now, these are fabulous fossils, but what are they? These creatures down here do not have any diagnostic feature that tell us whether they belong to the crown or to the stem. They are mysterious. And those creatures appear the radiation of the crown. Why did this happen around 800 million years ago and not hundreds and hundreds of millions of years earlier, as suggested by the molecular clocks? The amount of fossils that we have is not quite high enough to answer these questions. But maybe if we join these fossils, the body fossils, with a different type of fossil, with molecular fossils, we can collect more information. So let's look what these molecular fossils are. The cell membrane of algae and other eukaryotes contains sterols. We humans contain cholesterol. These molecules function as membrane rigidifiers. They modify the permeability and even the curvature of the membrane. Conversely, bacterial membranes, although not the membranes of all bacteria, contain so-called hopanols. If we are now in an aquatic ecosystem with water column at the bottom sediment, then our primary producers, algae and bacteria, when they die, they will sink very slowly through the water column, and most of it is recycled back to carbon dioxide and water. But very stable molecules like our sterols may survive and concentrate here in the bottom sediment. With more deposition of sediments over the top and the onset of reducing conditions, our sterols might lose their functional groups like the hydroxy group here and double bonds and turn into hydrocarbon scoutons. Such molecules, stable over hundreds of millions of years and containing still biological information, are called biomarker or molecular fossil. In the same way, our bacterial hopanols are turned into Holpane biomarkers. We can now grab a piece of sedimentary rock, remove contaminants, crush it to powder, fill it into metal bombs and extract it with hot organic solvents. What comes out is a brown liquid similar to petroleum and contains hundreds of thousands to millions of different molecules that we can analyze and separate using gas chromatography, mass spectroscopy. Using these techniques, we can then quantify and identify our different molecules. In this way, we can, for example, quantify stearanes and holpanes and compute the stearane-holpane ratio, which is a rough first-order estimate for the relative fluxes of algal versus bacterial organic matter to bottom sediments. In our fundamental tree of life, there is a lot of different biomarkers. In the eukaryotic part, there is 3 different stearane biomarkers that will be important in this talk. In red are cholesteroids with 27 carbon atoms, ergosteroids with 28, stigmasteroids with 29. They're distinguished by having no, 1, and 2 carbon atoms in the side chain. I will always show them in red, blue, and green. In front of a yellow background, I will show our bacterial holpanes. Now, all of these 4 biomarkers are not diagnostic for any specific group of organisms, but their relative abundances, their trajectory through time tells us something about broad evolutionary patterns. And that's what we're going to look at in the next slide. This diagram now summarizes the evolution of our red, blue, and green eukaryotic and the yellow bacterial signal through time. For example, the green color that you see here signifies the dominance of green algae in the Paleozoic and Ediacaran. If you would now go back in time to the Ediacaran, Caraginian, Antonian, all the way into the Archean, maybe you would expect more and more primitive and unusual biomarker signatures. However, if we take literature data from the 1980s to the present and place it into this diagram, then in cartoon form, it would actually look like something like this. This one looks more or less entirely like the Jurassic. And some 15 years ago, I looked at this and thought maybe all of this looks Jurassic because these signals are Jurassic. Maybe all of these are anthropogenic petroleum products, contaminants from drilling fluids, from cutting fluids, from diesel exhausts. So over the last 10 years, my PhD students and postdocs went back systematically through the entire Proterozoic, formation for formation, and reanalyzed it, using new techniques for the removal of contaminants from surfaces, looking through the veil of contamination and revealing a real Proterozoic biomarker signature. And that's what I'm going to show you in the next slide. Now here is an empty diagram, and I'm going to fill it in starting at the bottom and working my way up all the way to the Phanerozoic. Now, if you remove contaminants, it turns out there is no really indigenous biomarkers older than 1.7 billion years old, and there definitely are no Archean biomarkers. And I show this here by in gray. The oldest biomarkers really appear here at 1.6 billion years ago. And these rocks are beautifully preserved, full of biomarkers. And these biomarkers look like this. They're entirely yellow. You don't see a trace of red, blue, and green. There is no indication for any eukaryotes there. It's a 100% bacterial signal. And this purely bacterial signal persists all the way to the end of the Mesoproterozoic. Clearly, in the Meso and Paleoproterozoic, algae didn't play a role, and photosynthesis was firmly in the hands of bacteria, such as cyanobacteria. The really exciting thing happens here at 800 million years ago. 800 million years ago, we get the first real modern eukaryotic sterol signature. The signature becomes stronger. You don't only have red, you have a little bit of blue. We believe, following also Gordon Love and Roger Summons, that this is the emergence and rise of rhodophyte algae, of red algae, as primary producers in marine ecosystems. But the big bang of eukaryote evolution, of phototrophic eukaryote evolution, happens after the two snowball Earth glaciations, with the emergence of the red, blue, and green signature. This is probably the successive radiation of green algae, followed by so-called chlorophyll C algae, namely the dinoflagellates, coccolithophorids, and diatoms. That is a beautiful record, because what you can see here is the transition from a bacterial world to a eukaryotic world in two stages. First, the rise of rhodophytes, and then the rise of chlorophytes. However, this beautiful record also produces a number of problems, because I've shown you these things here, these clearly eukaryotic fossils throughout the late Paleo and Mesoproterozoic. These things are eukaryotic. So where is their membrane molecules? Where is their membrane sterols? Now the thing is, if we want to make this red, blue, and green signal visible in very low quantities, we need to tell our mass spectrometer exactly what to look for. If we don't tell the mass spectrometer exactly the right structure, it will not see it. There might be other molecules that are hiding in plain sight, maybe even abundantly, and we cannot see them. So maybe our early eukaryotes didn't produce red and blue and green signal. Maybe they produced different membrane lipids. To explore that possibility, we have to go again back in time. This time to the 1960s. In 1964, Konrad Bloch, together with Fyodor Lenin, won the Nobel Prize for deciphering the biosynthetic pathway of our cholesterol. The pathway he found out starts with this straight chain molecule here, squalene, 30 carbon atoms. Using the enzyme squalene monooxygenase and molecular oxygen, this enzyme produces epoxysqualene, And then the second enzyme, the oxidosqualene cyclase, is folding this molecule in one beautiful step into a molecule with 4 rings and an 8-carbon side chain. The product has 30 carbon atoms and is called lanosterol. Now the amazing thing happens. Using another 11 enzymatic steps, 12 enzymatic steps, and using a lot of energy and oxygen, this carbon atom, this carbon atom, this carbon atom are removed. The double bond is removed around until we end up with human cholesterol. Now Conrad Bloch noted that is a lot of waste of energy and oxygen to modify this molecule a little bit further. He called this the evolutionary perfection of a small molecule. And in a collection of essays, Blom's in Venetian paintings, the 9-banded armadillo, and other essays in biochemistry, Conrad Bloch made a beautiful hypothesis, namely that each of these sterols, once hundreds of millions of years ago, was a biosynthetic end product of an ancient creature that lived in deep time, and that the evolution of each successive step was an improvement as a membrane lipid over its predecessor. Now that is very exciting because it means if we go back enough in time, we might be able to find these molecules as the primary membrane lipids in deep time. Now, of course, everything since Conrad Bloch has become more difficult. For example, we now know that our blue ergosterols and green stigmasterols are not simply biosynthesized by sticking additional methyl groups to the side chain here. Instead, these two pathways have their own assembly lines, using much of the same enzymes, but in a different order. For example, in filamentous fungi, this methyl group here is already attached to the side chain in step number 2. But that is a great opportunity because we may find these molecules in very old rocks. So now we can go and take these biomolecules and turn them into molecular fossils that look like this. And we can look for these things in deep time. You already know that cholesterol here emerges at 800 million years ago, ergostein at 750, stigmastein at around 635. Maybe we can find these ancient molecules in deep time. And henceforth, I will call these molecules fossil protosterols. Before we set out on a quest to find ancient fossil protosterols, we need to figure out what sort of creatures may have been able to produce them. We know that our 3 sterols, the red, blue, and green ones, are distributed across the entire eukaryotic tree. We are therefore quite confident that LICA was capable of producing all 3 of them. Archaea, on the other hand, do not produce sterols. Therefore, the biosynthetic pathway from protosterols towards modern sterols must have been assembled along the eukaryotic stem, invented by stem group eukaryotes. To make everything a little bit more complicated, we also now know that there's at least 5 groups of different bacteria that can produce this type of protosterol here. And we know that the biosynthetic ability, the genes to do so, were horizontally transferred amongst those 5 groups. We also know that the 2 genes OSC and SQMO to make protosterol was horizontally transferred between eukaryotes and bacteria. However, we do not know whether these 2 genes evolved in eukaryotes and were then transferred towards bacteria, or involved in bacteria and were then transferred towards eukaryotes. In a sense, it does not matter. The group at MIT around Roger Summons found out that this horizontal gene transfer happened before 1.7 billion years ago. That is important because it takes us to the take-home message of this slide. If we find fossil protosterol in rocks less than 1.7 billion years old, then it could have a bacterial origin, for example, methanotrophs, or, and that's the exciting thing, it could be from stem group eukaryotes. So, my PhD student Benny Nettersheim set out to to find these things in the deep time record. When we first set out to find fossil protosterols in the ancient sedimentary record, we thought we'd be lucky if we find just traces of these compounds. But what we found instead really amazed us greatly, and you are going to be the first people that see these results. I will now add in dark gray our protosterol abundance data to this diagram. And the data is time binned and is stretched to fill the hardly considerable gaps in the geological record. What we found in the oldest sedimentary rocks looks like this. That is a lot of dark gray. In fact, the concentrations down there at 1.6 billion years ago are as high as the concentration of modern sterols in the Phanerozoic. Totally unexpected. And we now know that this signal is either from protosterol producing bacteria and stem group eukaryotes. That signal continues down here into the mid-Mesoproterozoic, but additionally you can see, down here that there is a little bit of purple. And that is 24-methyl protosteroids, and you know that that's a possible biosynthetic intermediate all the way up to the blue ergosteroids. But now comes something really, really important. Only eukaryotes are able to stick an additional carbon atom to the side chain of sterols. That gives us some confidence that at least some, or maybe most of the protosterol signal is in fact from stem group eukaryotes. That stem group eukaryotic signal persists to the end of the Mesoproterozoic, and then when the red signal here emerges, the dark gray signal starts to disappear. And then up here, all the way in the Phanerozoic, there's only traces of these compounds left. These small traces of protosterols in the Phanerozoic could be simply a contribution from some bacteria, plus the biosynthetic intermediates of protosterols found also in all modern organisms towards the production of the red, blue, and green signal. Now, that is a massive protosterol signal that we see in the mid-Proterozoic. And we do find these molecules in shallow water and deep water environments, in carbonates and in clays. We even find them in microbial mats and planktonic environments, and in freshwater and marine. I like to call these environments that are dominated by protosterols the protosterol biota. And it clearly made way towards a more modern sterol biota. Now we suggest that this is nothing else but the transition from a world that is dominated by stem group eukaryotes to a world where the bottom and the top of the food chain is dominated by crown group eukaryotes. And all of this really happened between 1,000 million and 800 million years ago in something which we may call the Tonian explosion of crown group eukaryotes. Now, I like this new data very much because for the first time, this reconciles our body fossil data and our molecular fossil data. Before, they told totally different stories. Exactly at the same time, when here our red modern sterol signal emerges, the same time our crown group eukaryotes start to radiate, that we know are descendants of LICA. Conversely, down here in the protosterol world, we have fossils of eukaryotic creatures that we cannot recognize as being part of the crown. So it's very tempting to say, well, maybe all or some of them are part of our earliest stem group ancestors, which evolved yet much deeper in time at a time when sterol biosynthesis had not yet evolved. And maybe this new data also helps us to answer one of the most difficult questions in eukaryotic evolution. You know that LICA, the last eukaryotic common ancestor, emerged sometimes between 1.2 and more than 2 billion years ago. But look at this. That means there must be a 400 million years to 1 billion year time gap between the emergence of LICA and the radiation of the eukaryotic crown. Where have our crown group eukaryotic ancestors been hiding for hundreds of millions of years? It's almost impossible to explain. But maybe the answer is hidden sometimes here in the Cryogenian and Tonian periods. We know that these periods saw massive perturbations of the carbon cycle. the emergence of the snowball earth glaciations, a surge in the concentration of the biolimiting nutrient phosphorus and also of trace elements. Maybe these changes, massive changes, were detrimental for our old world stem group eukaryotes that had evolved in a quite quiescent, stable mid-Proterozoic world. And maybe this gave crown group eukaryotes the edge. And this is my take-home message. Maybe just as dinosaurs had to go extinct to open ecospace for our mammal ancestors to radiate and evolve, maybe crown stem group eukaryotes had to go extinct so that our eukaryotic crown could evolve into empty ecospace. It would still have been very nice to inherit Stolberg Castle. Thank you very much. Scientific references and acknowledgements will appear in the following slides.